Okay, we are now live. Um, Hello, everyone. Thanks to those of you who I see we have about 20 people here. So those of you who have decided to uh, continue on this learning journey for 5 weeks, I feel like it's been fun and I've enjoyed it so far. I hope you all have as well, and hopefully we'll have a little time at the end of this last bit of content to maybe bring things full circle and have a bit of a discussion or Q&A if people are interested in sticking around for that. I'm going to um, share my screen here. And here I just wanted to show you at the, this is the workshop page. At the bottom is a link to a post-workshop survey. I'll copy that into the chat as well. Please fill it out whenever you get the chance. Hopefully today, and that will just give me a little information about how you participated in the class and what was good and what was bad about it, which will be very useful for me for future classes. It's a short questionnaire. So thank you in advance for doing that. Now, let's get to the content for today. I'll introduce some of the concepts and then we'll go and do a bit of coding shortly. So, as usual, we'll start by reviewing what we've learned so far and say how we're going to build on that. And you already know all this stuff. The protocol should be second nature to you by now. And as we've said many times, this is a course where we've learned some of the big concepts in Bayesian statistics, and through the last couple of weeks, you've been slowly adding different models to your toolkit of situations where now you know an appropriate Bayesian generalized linear mixed model to analyze that kind of data, and different experimental designs, different correlation in space and time, different structures or distributions of the response variable, including weird ones last week with zeros and that kind of thing. What are we going to do today? Well, these are both situations that I encounter often as ARS statisticians. So probably many of you have data sets that are like this. The first one is very common in ag science, especially in breeding, where you're phenotyping, where you have ordered categorical response variables. How do you analyze those data? And second situation we're going to look at is if you have a response variable that's continuous and nonlinear, and you have a specific functional form that you want to fit to that nonlinear trend. How do we do that in a Bayesian way with BRMS? Those are the things we're going to look at with examples that are appropriate for ARS scientists and the research they do. And a couple of more technical things that we're going to hit today as we do that is we're going to look in a little more detail at link functions, which we've talked about some. We're going to see some new and different ones. And we're going to look at how you would do a nonlinear mixed model, linear being intercepts and slopes, but what if you have nonlinear relationships? How do you incorporate fixed and random effects into that? We're going to look at that. So buckle up and let's go. These are, the first part is the cumulative logistic mixed model, which is the type of model that we're going to fit to ordinal data. I'll talk a little bit about what that is. We'll go and do that in a minute once we get to the hands-on portion. So I'll circle back to loading the packages. We'll talk about the kind of data we're going to work with. We've seen binary response data in lesson 2, like did the seed germinate, yes, or did it not germinate, no. That's a very common kind of data. what if you have more than two possible values? Well, there's two ways in which you could have data with more than two possible values. One would be a nominal type of data, like an unordered description. So just different categories, but there's no scale you can put them on. They're just different like colors or species or whatever are your response variable. Ordinal is the kind of data that we are going to work with today, where there's more than two, but they fall into an order, like very happy, kind of happy, not so happy, or like a scale from 1 to 5 and that kind of thing. There are statistical models for the unordered case, but we're not going to look at those today. Ordered categorical data. A lot of data in ag science follow this scale. It's very common if you have a complex phenotype that is very time consuming or even impossible to measure directly. You can just look at the organism and score it. Like if you have some disease severity, it's very hard to measure exactly. So you could just scale it from 1 to 5, or how hairy is this plant stem? Well, to get that quantitative, you'd have to sit there and count all those hairs, or you could kind of estimate it into some categories. And yeah, if anyone has worked with this kind of data before, put a thumbs up. I'm curious how many people here are familiar or have seen this kind of data before in their own research. I see that, at least half dozen thumbs going up. So hopefully this will be relevant to you all. Now, I often say, okay, here's the quick and dirty way, and now we're going to do it the right way. This is no exception. Obviously, it's necessary in many cases to get these categorical score variables because it's much faster. Obviously, it would be prohibitively time consuming to count every hair on a stem and that kind of thing. So you can phenotype a lot more organisms, and that's a good thing. But it comes at a cost, which is you lose a little bit of precision. You lose some underlying information, right? You've taken a continuous variable and turned it into one that only has a few discrete values. Not to mention there's an error associated with assigning something to it. You might assign some individuals to the wrong category as well and not even know it. So there's that error. And even if you did it perfectly, there would still be some information lost, right? So that means that you need more data points to get a similarly precise estimate as you would if it was a continuous variable. But some people want to have it both ways and do their quick phenotyping with a categorical scale, but then just pretend like that those 1 through 5 or 1 through 7 or whatever they are just continuous numbers and treat them as numbers and model them as having normally distributed error. That can work in many cases, and it can give you the right answer, but it can also give you an overinflated idea of how precise those estimates are. And we have statistical models for these ordinal response values, so why not use them, especially when we have the Bayesian approach with priors to help us constrain our estimates and regularize, as we've done time and time again in this course, right? If we have the tool at our disposal, and there's no excuse not to use it, I would say. So let's review, because this multi-category case with more than two categories is really just sort of a extension or a generalization of the binary binomial model that we've looked at before, right? Some people call it logistic model, because you usually model the binary data as following a binomial distribution with the logit or log odds link function. Why do we use log odds link function? Well, because the thing we're trying to predict is a probability, like the probability given the treatment will the seed germinate. That has to be between 0 and 1 because it's a probability. But our linear predictor, right, our term that's made of the fixed effects multiplied by the covariates plus the random effects, those can be positive or negative. It can be any number. So we use the logit, or the log odds function, which takes us from the probability scale from 0 to 1 to a number that can have any value, right? If you take p is a number between 0 and 1, you could get any number, positive or negative, depending on what p is, by taking the log of p over 1 minus p. That's the log odds, which works good for our generalized linear model. So if we had two only outcomes, binary data, we only need to predict one probability, right? If you know the probability the seed will germinate, you already know the probability it won't germinate, because those two things add up to 1. So you only need one number to predict two outcomes. So that's why the binomial distribution only has one parameter, theta. Like a coin flip, we can model it as following a binomial with one parameter, the probability of flipping heads. Simple enough. Multinomial distribution is what we're going to be working with now. The common thing you might be familiar with that follows a multinomial distribution is rolling dice. A dice roll can have more than two possible outcomes, right? It has n. A six-sided die has 6 possible outcomes. If you have 6 categories, you would have to estimate 5 probabilities, which all have to be, basically, they all have to add up to something less than 1, because if we know the probability of rolling a 1, 2, 3, 4, and 5, we add them up, and then we know the probability of rolling a 6 as well, because those are the only possible outcomes. So that's a multinomial distribution. If we have n categories, it has one less number of parameters than that. If that makes sense to you, can you give me a thumbs up, please? Or if you are confused, feel free to ask a question. Okay, cool. So instead of dice going from 1 to 6, our outcome variable is some categorical score of a phenotype going from 1 to whatever. We use a similar logit kind of link function in the multinomial case, but it's a little more complicated. Like, let's say we had 4 categories. We're going to have to estimate 3 parameters, right? The probability of the outcome being category 1, the probability of it being 2 or less, and the probability of it being 3 or less. And obviously, since there's only 1, 2, 3, and 4, the probability of 4 or less is 1, so we don't have to estimate that. And so those are sort of cumulative, 1 or less, 2 or less, 3 or less. They're always higher. One is higher than the next. And so that's called cumulative logit model, is what we're going to fit. And you don't need to worry too much about the equations, but the cumulative logit model has not just one link function, it has n-1 link functions, one for each of the thresholds between the categories. And it, just like in the binary case, it takes a cumulative probability, which has to be between 0 and 1, and maps it onto a scale that can take any value. So this first one would be like the probability of getting 1 or less, which is just the probability of getting 1. Then here's the probability of getting a 1 or a 2. And oh, these should just be equal signs, not less than or equal to. So I'll change that notation later. So that's the log odds of getting a 1 or a 2, getting a 2 or less. Make a note to change that later. Like I said, I haven't used these slides before, so they might have a few mistakes in them. Here's the example data we're going to use. This is also from the AgriDat package, where I've gotten many of my examples from before. This is a data set published by Karcher et al., where they wanted to know what way of applying nitrogen fertilizer and how much of it affects turf grass color. I guess in the world of turf grass, color is very important. You want a nice looking sod. So they were concerned with how different fertilizer regimes basically affect that. And this is a completely randomized design. There's no blocks in it. They just had two different treatments. One was sort of the management treatment. Where was the nitrogen applied? At the surface, or was it injected at three different depths? So surface, depth 1, 2, and 3. There's 4 levels there, and they did low and high amounts. 4 by 2. There's 8 combinations. They were applied to little patches of turf in a completely randomized way. There was no blocks. And they rated the experimental plots for how nice they looked, what the quality of the color was for 4 weeks. And each week, each combination within each replicate was rated a variable number of times on a scale. Now, this is the important part. The scale had 4 categories, one being better than the other: poor, average, good, and excellent. So excellent would be like a nice bright green, I guess. And but there's no sort of continuous variable that corresponds to this scale, right? We couldn't say, well, excellent is 5 times better than good, but good is 2 times better than average, or anything like that. We don't know that. All we know is that excellent is better than good, is better than average, is better than poor. That's all we know. So that's why we can't translate that into some continuous number and fit a model as if it was a numerical value. We shouldn't say it's 4, 3, 2, 1 and fit the model accordingly. We need to use this cumulative logit to treat them as discrete ordered categories to get the most accurate inference. So without further ado, let's go and into Scinet or into RStudio on your own machine and get a session going so that we can start looking at this data and start fitting models to it. So if you remember, if you go in, I've already logged in to Scinet and started a session. But if you go in and go to the My Interactive Sessions tab, you can go to your last week's server session that's still there and click on this refresher, or which will start a new session with the same parameters as last time. But if you don't have that for whatever reason, you would go to RStudio server and, as usual, start a session under SEA stats training, series, partition, R 4.4, with an adequate number of hours, cores, and memory. And you can use this reservation equals base 2025 as we've done every time. So hopefully you all should be able to do that by now. And once you start that, you go to launch RStudio server, and it will open up this page in your browser with the RStudio console. And I've gone into the worksheets tab here, or I'm sorry, the worksheets folder in the files tab, and open up the lesson 5 worksheet. Hopefully everyone is able to do that. Just to make sure everyone's good, can you give me a thumbs up if you either got there or you're about to get there? If anyone is unable to, for some technical reason, please let me know.
Yeah, Quentin, since this is the first time I'm going in
Yes.
on the series rather than on my own, what was the optional slurm argument again, please?
Oh, you don't actually need it because it's optional, since I reserved some of the nodes on the Slurm cluster for our use of our of this class, so it just guarantees that you'll get some processors to use. But even if you don't put it, you'll probably get some. I mean, it's unlikely
so
they're all being used. You can just go for it even if you didn't type that in. But Grant put it in the chat as well.
Thank you.
But after today, that reservation will never work again. So definitely delete it from your if you're ever going to use SciNet anymore, even for this SEA stats training account, you're not going to be able to use the Bayes 2025 reservation anymore after today, since it's only valid for today. But that's okay, because it just means your job will go into the main queue, and usually they start very soon anyways, unless, it's somebody's like bogarting all the cores for a huge analysis or whatever. Yeah, so here I am. I've opened up the worksheet, and let's go ahead and, as usual, run the first lines up to this part. Hit Control Enter. That configures everything and loads all the packages. Then we can run line 28. I'll put my cursor there and hit Control Enter. And that will load the data set that is built into the aggredat package. And then on the next line, I'll hit Control Enter line 34 just to take a look at the first few rows of the data set and see what's in there using the head function. You can see there are time points, weeks from 1 to 4. Reps are the replicates. It's a completely randomized design, so those are not necessarily blocks. Like everything from rep 1 is not in one physical location, so we don't need to have a random effect for rep in our model necessarily, or we should not, because it wouldn't make sense to. Management has those 4 levels. They apparently represent different depths of application. Nitro is 1 and 2. That's low and high fertilizer application levels. And rating, you can see it has poor, average, good, and excellent. So it's a text value. And this is the number of ratings of that rating class for that particular replicate in that week. And let's look at the distribution of rating categories in the data by using the table function, Karcher.turfgrass $rating will take the rating column from that data frame and make a table of how often each value occurs in that column. Control Enter to run that. So you can see, first of all, the ordering of the values is in alphabetical order by default. If you loaded this data from an external source, R would not know off the bat that excellent is supposed to be the highest, then good, then average, then poor, right? It orders things alphabetically by default. So we're definitely going to have to tell it how to order the factor levels so that we get the correct answer later on. And also note that only one of the many values is excellent. So here I would say, if this was a data set I was given, I would say we probably aren't going to have enough data to distinguish the effect of those 8 different treatment combinations on the probability of being excellent, because only one treatment combination at all must have had any excellent values in it. So what I would recommend doing in this case is pooling that value with an adjacent category. Again, we're losing a tiny bit of information there, but I don't think we're losing much, because, as I said, there's not enough information in the data to say anything about the probability of excellent. So what I'm going to do is I'm going to pool good and excellent together and just call it good plus, like good or above. And that'll be, so we'll have 3 categories: good, average, and poor. And this next bit of code is to process the data. So first, we're going to mutate the rating column by first pooling good and excellent together to this good plus value, and then setting it as an ordered factor. So rating is the name of we're just going to modify the rating column in place and change the value in that column. So rating equals if else parentheses, rating in good or X. So if rating is equal to good or excellent, we'll turn it to good plus. Otherwise, we'll keep it as is. Then we'll pipe that result into an ordered factor, which we use the ordered function to create an ordered factor. That's a special data type. And levels equals poor, comma, average, comma, good plus. So we've specified the ordering from worst to best, from lowest to highest of that factor. Then you can see that the way that this data set is structured in the example data set is that it's giving us a count for every treatment combination and every rating, but the way that BRMS expects data for this kind of model is instead of having a count of 2 here, we would want 2 rows that said average, average. And here there would be 3 rows for good, good, good. So we're going to, the next few lines of code basically move the data into long form. So like you see that this currently has 128 rows. We want 1 row for every single observation. So the number of rows is going to be the sum of all the counts we have. And the way we do that is by grouping by the different combinations and then reframe, which means create a summary, which may be variable number of rows for each group, and use rep rating, comma, count to repeat the rating value that many times. And we're also going to, while we're at it, turn week into a factor variable to treat the time points as discrete. So all of that is our sort of data processing pipeline to get the data into a format that we can work with. I'll hit Control Enter somewhere on there to run that. Now you see Karcher underscore long has 250 rows. We can look at it in here. You see rating is an ordered factor and everything else is a factor. So there's no continuous numerical data in here. It's all categorical. Any questions on that so far? So these are just kind of standard processing things we have to do when we want to input a data set into a statistical model. And of course, these example data sets from AgriDat are already very clean. They're just not exactly in the format that any particular statistical program wants. In real life, you'd have to do even more data cleaning than that. Let's take a look at our data by plotting it. We're going to use our x-axis in the plot is going to be interaction parentheses nitro comma manage. So the x-axis is going to have 1 space on it for each of those 8 treatment combinations. There's 2 levels of nitro and 4 levels of manage. And we're going to make a bar plot with different colored sections for each rating value. And stat equals count. We're going to count the number of ratings we observe in each treatment combination. And position equals stack. We're going to make a stacked bar plot with the poor, then average, then good, then good plus, I guess. And we'll make a separate one for each week. Control Enter to generate the plot. So here we see, let me zoom that in a bit so you can see it better. Here we see that there's clearly some differences between the treatment combinations. This is on the left, we have our M1 treatments, whatever that is, surface application of nitrogen. Whether it's low or high, we have pretty much poor to average ratings. Same with the M2 treatment. The M3 and M4 treatments seem to be a lot better, right? They have mostly good, not very many poor and average ratings. But there's maybe still an interaction with treatment with the N and the M treatment aren't exactly the same. The high nitrogen, the N2, tends to have more good ratings. And is there a trend over time? You tell me. I think each of these 4 panels represent different weeks. Maybe there's a modest trend over time, but I would say for the most part, the relative differences between the treatment combinations are similar-ish over time. But we can fit a statistical model to formalize our intuition about the data that we get from looking at the plot. That's our bread and butter, right? So that's what we're going to do here. So let's take a look at the syntax for the cumulative logit mixed model, which I call CLMM for cumulative logit mixed model. And rating, our ordered factor, is our response variable. I am going to, here, just to simplify things a bit, I'm not going to fit all 3-way interactions. I'm going to fit nitro asterisk manage plus week, not asterisk week, because I don't think there's a very strong interaction between treatments and week just from looking at the data. So at least for now, we're just going to exclude that from the model. I can't remember if it's an exercise or if we're going to do this in our example here to include that interaction and see if it changes the model fit. But in any case, for this, just to simplify things a bit, I've excluded that interaction. Now, I said it's a completely randomized design. Yes, we don't need to put a random intercept for replicate, but we do need to account for the random, for the repeated measures that were taken on the same individual plots for 4 different weeks. You could do that either by putting in a residual covariance structure like we did in week 3, or kind of an alternative way to do it that does not account for the time structure but is decent enough for our purposes is just to fit a random intercept to each plot. And that's what I'm going to do here for simplicity's sake. And I think it will fit the data quite well. So we're going to, each plot is identified, right, by a unique combination of the replication, management, treatment, and nitrogen treatment. There's only one plot for every unique combination of replicate, management, and nitrogen, because each replicate has a complete set of all the treatment combinations. So to identify that, we are going to put, not there, we're going to put it here, rep, nitro, manage. That could be in any order. It doesn't matter what order you put them in, because this is just making a new variable that is the unique combinations of those 3 columns. And the 1 means we're doing a random intercept. The family is how we're going to tell BRMS that it's a cumulative logistic model. The family is called cumulative. And link equals logit. Again, that's the default, but I'm typing it in here anyway. Another default that I'm just including to be explicit is threshold equals flexible. And what that means is we are fitting a separate parameter for each threshold between the categories. If we didn't, it would mean that they would be equidistant apart. But that is not necessarily the case. Flexible is, like it says, more flexible. If you could play with that and see if you get adequate model fits by forcing the thresholds to be equidistant, which does have less parameters, it could be a more parsimonious fit in some cases, but I rarely stray from the flexible on default. Okay, and then as always, the wizardry of choosing the right prior, as usual, because this is a log odd scale model. Small numbers for the standard deviation on the prior is fine, because on the log scale, even effect size of 2, 3, whatever is a big effect. So I'm going to put normal mean of 0. Again, we're not saying one treatment is higher than the other, and we're using a standard deviation of 2 in this case, since that's reasonable for the log odd scale. And class equals B because we're putting that prior on the fixed effects. And that's that. Other than that, we're using default model fitting options. I'm going to hit Control Enter and run that. Again, because I did not make any changes to the model or the data. Since it was last fit, the pre-fitted model object loads automatically. Everyone good there? Or if please ask if you have a question. Cool. Let's do a posterior predictive check plots just to quickly see if the proportion of values in each of the categories predicted by the model is roughly similar to what is observed. So we're going to use that posterior predictive PP underscore check. The name of the fitted model object is the first argument. We're going to use bars underscore grouped as the type of check plot, right? Because if you don't specify this by default, it'll give you that continuous plot, which doesn't make sense for this data set because it's a discrete thing that we're predicting. And first, we're going to look at it two ways. First by management, then by nitrogen, just to see if it roughly reproduces the relative proportions in the data. So let's take a look. So here you see that it looks pretty good, right? The blue bar plot is the counts in each of the three categories that is in the data. Okay, Marina, the command stand path has not been set yet. So yes, you're going to need to, sometimes, I don't know why that happens. So you're going to need to run, you're doing this on Scinet and not your own computer, right? If it's on Scinet, you can just run that line and it will set the path. So try that and see, then run that source.r profile and then try again. Let me know if that works. While she's trying that, just to reiterate, the blue bars are the proportions in the categories observed in the data. The dark blue dots with error bars are the range of predictions from the posterior predictive. We can see that it's very close match. And let's look at it for nitrogen as well. Hitting Control Enter on line 59 here. Okay, great. Glad that works. And again, we see a reasonably good match between the data observations and the predictions. So all is good there. There are various ways, if you didn't see a good match, that you could relax some of the assumptions made by this model fit, but we're not going to get into those here. Yeah, so before we go and try to make formal predictions and things that would be manuscript quality, I forget if I've shown this function before in this workshop, but BRMS has a built-in way to just quickly look at the fixed effects in your model, the predictions, without doing anything real fancy, which is the conditional effects function. If you just have a regular model that's not some weird categorical model, you can just run conditional effects, parentheses, your model, and it'll give you a bunch of default diagnostic plots. In this case, we have to supply some extra arguments to make the plot that we want, but I'll just show you that here. So this is kind of a quick diagnostic to look at the fixed effects in our model. Conditional underscore effects is the name of the function. The first argument is the name of the fitted model object. We have to put categorical equals true to specify the response variable is categorical. And then I'm specifying which effect I want to see the effect of management across the two different levels of nitrogen. So we're going to see all 8 combinations here. What is that going to look like? Let's take a look. Hit Control Enter here. So here you can see that we have 8 treatment combinations. So this left panel is nitrogen level 1, the low nitrogen level. The right panel is nitrogen level 2, high nitrogen. And within each one, we had 4 different ways of applying the nitrogen. But here's the thing. We don't just have we don't just have 8 responses. We actually have 8 times 3, right? Because for each of those treatment combinations, we've estimated 3 probabilities: the probability of being poor, probability of being average, and probability of being good. So that's actually what our model is predicting back at us. Like, for example, our model is predicting us that if you are at nitrogen level 2 and management treatment 4, you have, over a 90% chance of having a good rating and 5% of being average and almost 0% of being poor, right? Versus if you have management level 1 and low nitrogen, you have about 50 chance of being poor or average and almost no chance of being good, and so on and so forth. It's actually quite confusing sometimes to interpret the output of a categorical model like this because of all the different probabilities you get. So you have, however many treatment combinations you have times the number of categories you have. That's how many different things are being predicted. So that's basically how you fit one of these kind of models. It's pretty straightforward. The rest of the time for this example is devoted to messing with the values of the posterior distribution to make different predictions, compare the levels, compare the means from different treatments, and make inference off of them, as we've been doing with every model we've done so far. In many of the cases, we have used the EM means package. Unfortunately, as it stands right now, the EM means package does not have the appropriate methods to automatically give us predictions from a categorical, a multinomial model like this. But we can take a slightly long, we don't have those shortcuts available to us, but we can use the tidy Bayes package and kind of make the predictions and manipulate them without using EM means. It just takes another extra step or two. So what we have to do is create a grid of combinations of predictor variables of fixed effects that we want to make our predictions off of, which I'm calling pred grid. And I'm going to use the expand underscore grid function to make this grid. And what this is doing is it's taking out of the Karcher long data frame every unique value of nitrogen, so the 2 different levels there, every unique level of management, there's 4, and every unique value of weak, there's also 4, and giving me a data frame with every possible combination. So 2 times 4 times 4, 2 times 16, there's going to be 32 rows in this data frame. Control Enter to run that. So pred grid, you can see here, has 32 rows. Now we're going to use the function add underscore epred underscore draws to, for every one of those combinations, get our posterior predictions of the probability of each category. Basically, the thing we're looking at in this plot right here. But we have to specify RE underscore formula equals tilde 0, meaning we want to set all the random effects to 0 for the purpose of making these predictions. Since we want, usually when we make inference, we want to make, and this is what EM means is always doing by default, you just don't usually see it. It's setting those random effects to 0 because we want the expected value for sort of the average plot of turf grass in that treatment combination. The average one that's smack dab in the middle. That's what EM means is always doing by default. And so that's what we're doing here. And then category equals rating means we're going to name the resulting column rating, which will say which category everything is in. I'll hit Control Enter. Karcher underscore epred. Now you can see this thing has 384,000 rows in it. What all is in there? Well, you can see, right, we had 32 treatment combinations, And remember, we had, by default, we got 4 Markov chains that run each for 1,000 sampling iterations. So that's 32 times 4,000 already. And then for every posterior sample, it's not just one value, it's 3. It's the probability of being poor, average, or good. So that's 32 times 4,000 times 3, which I guess is 384,000. That's how many numbers we just churned out. That's the full posterior distribution of all these probabilities here. Make sense? Okay, so what are we going to do? Well, let's say that, again, because we didn't really, at least for the purpose of this simple analysis, we're not going to care about the trend over time. So what we can do is basically just, and this is also what EM means does internally, but we're just doing it manually here, is just average each one of those predictions across the levels of weeks. So we're going to take this data frame we just created. We're going to group underscore by nitrogen management. This dot draw is a column that is created by the add epred draws. That's going to be 1 to 4,000 for our each iteration of the posterior. And we're going to group by each rating category as well. And epred is also by default the name of the column with the predicted value with the probability in it. We're going to take its mean. So we're going to average our predictions over the 4 weeks for every treatment combination that we have and every rating level for each posterior draw. We're going to average over time. And then what I did here is just attach that average prediction averaged over time back to the original one that had each individual week, so we can make a plot that has the individual week predictions plus an average over time prediction. That's what I did there. And now I'm going to make a plot. I'm going to make a plot that shows this. So now this is like a slightly more customized plot than the quick diagnostic plot that you would get from BRMS. So let's go ahead and do that. And so we're going to, as before, the combination of nitrogen and management is on the x-axis. The y-axis is this dot epred, which is just the default name of the expected value of the posterior predictive. So that's the predicted value from the model. And we're going to group by rating. So we're going to have a separate bar for every rating class. And then this stat interval is some code that will make a summary statistics off the posterior distribution using its quantiles. And then I'm going to also put a point for the median of each posterior on there. So fun in the stat summary, fun equals median, and geom equals point. And then we're making a separate panel for each week. So this is a fairly complicated plot. It's going to have a lot going on. So we'll generate that plot hitting Control Enter. So yeah, this is kind of like all the results, but look how much is going on here, right? There's you know, this is kind of the messy reality of it, right? We have a lot of treatment combinations, we have different time points, and for every single one of those, we have 3 different numbers we have to look at, right? The probabilities of each of the rating classes. You know, there's clearly effects of the treatments here, but if you were looking at this plot, it would take a while to unpack, I think, like if you saw this in a journal article or a slide or whatever, or where you're looking at it right now. So there is kind of a way in which people simplify these results down, and I'm sort of ambivalent about whether it's a good way or not, but it is a way that you often see, and I've used it myself as well. So I'm going to show you how to do it as well. Basically, we're going to take these 3 numbers for each treatment combination, right? The, you know, Here we have a low probability of poor, a pretty high probability of average, and a low probability of good. So like we could say, well, this treatment combination here pretty much yields, for the most part, average colored turf grass, right? In this particular week. So let's go ahead and distill that down into one number, basically by going back in and treating each of these rating classes like it is a number, and then taking the weighted average. Like if we said poor is 1, average is 2, and good is 3, we'll take a weighted average of 1, 2, and 3 weighted by those probabilities, and we'll probably get a number pretty close to 2. And that's what they call the mean class prediction for that particular treatment combination. So if we do that, we can highly simplify the numbers in this plot at the expense of losing some of the information that's in there. But I'm going to show you how to do that. So basically, we go back to our predicted values we generated, and we're going to group them by all the treatment combinations by each posterior draw, but we're not going to group by rating anymore. We're going to basically, instead of the 3 values for each combination, we're just going to have 1 now. And we're going to make a column called mean class by using the weighted dot mean function. And x, the thing we're taking the weighted mean of, is as dot numeric parentheses rating, which will by default turn poor to 1, average to 2, and good to 3. And w equals EPRED. We're going to weight the average of 1, 2, and 3 by the probabilities of each rating class. So like if you had 100% probability of 3, the result would be 3. But if you had 50-50 between 2 and 3, you'd get 2.5. That's what you get there. So I'll hit Control Enter there. Because you see, it's not perfect, right? Because if you had 0 probability of poor, 100% probability of average, and 0 probability of good, you'd get an average of 2. But if you had a third, a third, and a third, you'd also get an average of 2. So you're losing a bit of nuance when you do this. But it does make things a little more, it makes the message simpler. But right, you know, Einstein said everything should be as simple as possible, but no simpler. So you have to weigh whether this is appropriate level of simplification for you. So let's make a plot of that. This is very similar to the code we had before, except the y column is now called mean underscore class. Mean underscore class. And I'm just going to use a blue color scheme anymore because we don't need 3 different colors anymore. Control Enter. So now here's like a bit of a simplified, dumbed down version of the one we had a minute ago, right? So now it's like, oh, here's the average rating class for this particular treatment combination. Like in week 1, the nitrogen 1 management 1 has an average 1.5 rating. So it's like between poor and average, versus the nitrogen 2 management 3 has an average of nearly 3, or almost 100% probability of getting a good rating there. So that can be a little easier to interpret if it still captures what's going on in the data. Thumbs up if you're following this. Okay, some people are paying attention. Excellent. Now, yeah, just a little bit more on this example. We'll take a break in just a couple of minutes. I also wanted to show, obviously, we've made this plot. People might also want to make inference about the difference between the treatment combinations. Well, since we have a Bayesian model, all you have to do, since we have a posterior distribution of the means for all the treatment combinations, we can just difference whatever pair of treatments we want to compare, and then look at the posterior distribution of the difference. That's what we've been doing all along. That's what EM means is doing by default. That's what we're going to do now in a slightly more manual way. But actually, we still have a shortcut because tidy Bayes has a function called compare levels that allows us to do all pairwise comparisons between particular levels. So we don't have to actually manually go in and subtract all the differences. So what I'm going to do is, first of all, let's just, for simplicity's sake, let's say we're just going to not do this for each week separately. Since we didn't look at an interaction with week anyway, we're just going to look at the overall averaged over time difference between the treatment means. And again, we're going to look at the difference between these mean class predictions, even though it's a bit of an oversimplification. Although you could do this separately and give odds ratios for every pair of treatments for every rating class, which would be the more honest but also more verbose way of doing it. So first, I'm going to just filter by week is equal to overall. We're just going to look at the overall, not look at each week separately. And I'm going to use the compare underscore levels function to compare each level of, we're going to use the argument variable, which thing are we going to take the difference of, which is the mean class prediction, mean underscore class. And what are the levels that we're going to compare to each other? By equals manage. So we're going to take the contrasts. We're not going to do the interaction. We're just going to look at the difference between each level of management, 1, 2, 3, and 4. Those are the contrasts for management, the 4 different levels that we had. Control Enter, and we can do the same thing for the low versus high nitrogen by just putting by equals nitro instead. Same code otherwise. I'll hit Control Enter there. Then just like we have done in previous lessons, we can use the describe posterior function to get credible intervals and Bayesian style p-values on those differences. But we have to do some extra group and summarize steps because, again, we're not doing this automatically within EM means. We're doing it more long-handed. So we want management contrasts by nitro and manage. So within each overall average over time, within each level of nitrogen, is there a difference between the management levels? And we can use the describe underscore posterior function within summarize because we have to do this sort of more longhand. And the name of the variable we're summarizing is this mean underscore class difference. As usual, we're getting some standard credible intervals and different Bayesian p-values. Hit Control Enter to take a look at what we get. Here are the, so basically within low nitrogen, is there a difference between management treatment 2 and management treatment 1? The difference between their mean class predictions is only 0.2, so there's not much evidence for a difference. But is there a difference between management 3 and management 1? There's almost a 1-point difference, like the difference between poor and average, or the difference between average and good is 1. So it's like one whole unit difference. And there's a pretty narrow credible interval on that. So there's very strong evidence for a difference there. And we can do the same to look at the difference in ratings between low and high nitrogen fertilization levels. Here you see that, regardless of the management treatment, the difference between high and low, high nitrogen always has a higher mean than low nitrogen, right? These values are always positive, and we have variable levels of evidence for how big of a difference that is based on the credible intervals and the Bayesian p-values. And in the slides and in the full text of the lesson, I have included slides talking about how you would present the text in the methods section and in the results section as usual. And if you did this analysis and wanted to put it in a manuscript, I won't go over it here, but you can look at it. You can look at it on your own. But I did include the code to make a slightly nicer figure. And again, so I've dispensed with looking at variation over time, but I have, instead of using the slightly sketchy predictions where I've converted things to 1, 2, and 3, I've kept it as probabilities to kind of be more closer to the data and not to this abstraction of turning it into 1, 2, and 3, and kind of that makes a lot of assumptions. Like if you do turn it into 1, 2, and 3, you are saying 3 is exactly 1 unit better than 2 is exactly 1 unit better than 1. But what if like poor is terrible, but average and good are not that different from each other? That distinction is lost if you turn them into 1, 2, and 3, right? So this figure here goes back to the probabilities. I've made a separate panel for poor, average, and good, which I think is kind of a nice way to look at it. And it is, since I've split it up a bit, it's a little more interpretable. So here's your low nitrogen. You can see that, you're very likely to have poor color in these two management treatments, versus in high nitrogen. In these two management treatments, you're still more likely to have poor color, but not as likely as if you had only low nitrogen, and so forth. That's what I have about multinomial models for categorical data. Does anybody have any questions for me about that? The question from Alan, can that last concern be mitigated by how you set up your scale? Which concern are you referring to? Sorry.
Yeah, sorry. No, you were just talking about the fact that poor against the poor could be relatively far away from average and good could be relatively close together. But is that not partially a function of how, when you're setting up your experiment in the first place, how you set up your scale?
Yes, that's true. Nothing's stopping you from like saying, okay, we're going to give poor a value of 1 and medium a value of like 8 and good a value of 10. And then
right.
You could certainly do that, and nothing's stopping you from doing that, and that would be a fine way to mitigate that problem. But again, it still wouldn't erase the issue of like if you had like kind of, sometimes you have sort of like a U-shaped distribution where like you have a lot of poor, not very much medium, and a lot of good. That is where this type of model breaks down a little bit, right? Because that would look like it's average, but it's not, right? So
got it.
If you have situations like that, you have to modify the model accordingly, because this model is not intended to deal with those type of situations. It's intended to deal with kind of linearly increasing probabilities, basically. So yeah.
Okay, yeah, that makes sense. I guess my point is that some of this is a function of how you choose to set up your experiment in the first
Very
place.
true.
And if you're considering what statistical approaches you're going to use to analyze it afterwards, then you can tailor how you gather your data.
Yep.
In
And
many
I
cases.
think it is probably, especially when you're doing these categorical scales, it's very important to validate those scales ahead of time by having at least some data where it's some samples where they are compared to the underlying value they're supposed to be approximating, to make sure they actually do correspond to something in reality. So, I think, and also like you'd want to have multiple raters do at least some of the phenotypes on the same individuals to make sure that the rating scale is consistent across raters and stuff. And there's statistical protocols for like seeing how consistent they are and stuff like that, which
sure
I won't get into. But that's very important.
I've actually used that before, where you train the raters in advance and make sure that you're getting consistent ratings, or as best you can with the human error. But yeah,
that's right.
Cool.
Just like you would calibrate any instrument you use to measure things. No difference there. Yeah.
Exactly right.
Very cool. Well, any other questions? Right on. Well, let's take a 10-minute break and come back at 3:15 17 Eastern. I'll be here for most of the break to chat. But I will step out for just a moment, and I'll be right back. We'll start again in just a minute. Okay, so hopefully everybody had a good break, and now we'll get started on the second example, the second and last example of this entire 5-week odyssey, which is we're going to look at fitting nonlinear models with BRMS. I'll go and share screen again. Nonlinear mixed models, the last, kind of unrelated to what we just did, but another thing that I think is relevant. You can tell it's relevant because I got the example data from a data set that I worked with a ARS scientist on. So clearly it's something that at least some folks in this area work on. We've kind of looked at some nonlinear stuff before, once in this set of lessons, which was 2 weeks ago when we were looking at spatial variation. We used what was called a generalized additive model, a GAM, to model the spatial variation, right? Because spatial variation is not always linear. Some environmental factor doesn't just go up or down on east to west or north to south. It kind of varies in a weird way over space. So that's why a GAM is like a semi-parametric model. It's a good model to use. It basically approximates a nonlinear relationship by sticking a bunch of polynomials together, which are linear models. It makes a wiggly squiggly line by putting a bunch of little polynomials together. And that works great for a lot of nonlinear relationships. Yeah, you could use it in many situations and it will be fine. But what if you have a strong theoretical expectation about how two variables must be or should be related, right? A GAM is semi-parametric. It doesn't really give you parameters that you can point to and say, hey, this parameter of some equation is higher in this treatment versus another treatment. So this treatment is having an effect on the growth rate of a microbe, which is some parameter of a nonlinear growth curve. What if you really wanted to be able to say that? It's kind of hard to say that with your squiggly wiggly lines, but if you have a true nonlinear function that you think things should follow, well, why don't we just fit that nonlinear model? And of course, BRMS, being the versatile piece of software that it is, has the ability to do that. And we can, all the little fun frills that we've talked about before, we can still do. We can put any error distribution we want on there. We can put fixed effects on the parameters. We can put random effects. We can do anything we want. The sky's the limit. I know you're as excited as I am about that. One, I just mentioned microbes already, so I gave it away a bit, but that's what we're looking at here. You probably know this from some pretty fundamental theory that if you put a bacterium on like a culture plate, it's going to start dividing. It's going to grow, because 1 divides by 2, 2 divides by 4. It grows exponentially at an increasing rate until after a while it runs out of food, or it starts to crowd each other out. It's drowning in its own poop. Then the growth levels off. You get this S curve, right? But a lot of mathematical functions look kind of like an S. There's not just one S curve function. There's several different ones. You might have, for a particular situation, a hypothesis about which functional form, which special S curve sigmoid function fits the data best, based on some knowledge and theory. And so that would be like, even if you weren't doing it Bayesian, deciding which function to fit is part of those prior assumptions and prior knowledge that goes into model building. It's Bayesians do it as well as even if you were doing it frequentist, you would also that would also be part of your priors in a way. Here's one of the many, probably one of the most commonly used S-curve functions that's out there, the Gompertz equation. And, it's just some letters on a page, but it's been shown empirically a lot of times that it is a good model for the growth of microbial biomass. So it's used for a reason. You see, it's got variable t in here, time. F of t is the population size as a function of time. And it's got you have your E, and you have three parameters, A, B, and C. A is basically the carrying capacity of the environment, the asymptotic population size. That's like how high up the S gets before it flattens off. B is sort of an offset, moving the curve forward or backward in time. It's the length of the lag phase. How long did the bacteria kind of slowly build up before they start to shoot up? And the C is the growth rate. Once they start to shoot up, how fast did they hit that carrying capacity? Jesse, I can see your screen right now. I don't know if you meant to do that. There we go. Yes, and that's the Gompertz equation. Here's a couple of graphs of what it looks like if you modify these different parameters. Here we've held B and C constant. No worries, I just didn't want you to accidentally show something. You hold B and C constant, and here's some different A's, right? So as A increases, your carrying capacity, the ultimate level of biomass increases. B, as you increase B, the length of the lag phase increases. Like if B goes all the way up to 100, the bacteria sort of take a long time to go to this exponential phase. And C, if we hold A and B constant and increase C. Oh, I didn't share my screen. Oh, yeah.
No, you're resharing. Yeah.
Thank you for telling me that. I've been going off slides this whole time. I see. That just happened just now. Okay. Good. So you were able to see my slides before, right? Just now is when cool.
Yes.
Excellent. So yes, let's see. I don't know when that started, but basically here's different A values. The higher they get, yeah, don't worry about it. These things happen. The higher they get, the higher the carrying capacity. The S curve stops at a higher level. B, the higher it gets, the longer it takes to start growing. And C, for different values of C, you get different slopes, different growth rates. So like 2 is a high, it goes up really fast. 0.5, it doesn't go up as fast. Those are the parameters. Everyone good? We're all good now? Thumbs up if it's all good. Okay, excelente. Well, here we're going to just introduce the example data that we're going to fit this Gompertz curve to. I extracted it from a real data set. I've taken away some of the treatment combinations that would just confuse you, and I've thrown in some noise as well. Basically, this experiment was set up to learn how much compounds called pyrosidines inhibit the growth of a very damaging pest fungus called Fusarium. And this was done using this little machine called a bioscreen, which is basically you stick a growth plate, a 96 well plate inside of this machine, and it can measure the growth of this mold basically under different conditions. And there were three replicates, 96 well plates used, but I don't have all of the treatment combinations in the example data set. But there's basically two compounds. They were trying to look at how well they suppress the growth of this fungus, A and B, and then to see if they interact, added A and B together. There's no control group with neither A nor B added. We just have A, B, and A plus B are our 3 treatments. And basically, this machine automatically measures how much growth has taken place in each well at regular time intervals. And how does it do that? Well, you can't really sit there and weigh the biomass of the microbes in the wells, right? They're suspended in some kind of fluid. So the way it works is you shine a bright light through each well and measure how much light is blocked by the stuff that's in there. And as it turns out, that number called optical density is a very closely related to how much biomass is in there. So the more light that's blocked from going through the well, the more cells are in there, meaning the more population is in there. We can pretty much say optical density is a proxy of population of mold in the well. So that's what the thing is actually measuring, if you're curious, called OD. Okay, so now let's go in and start exploring this example data set back in our studio server. Here I'm down on line 129 here, although your numbers may vary by now. If you're on Scinet, we can just run line 29 and import this file from the data folder. If you're on your own machine, I've posted the CSV on a website that you can load. Okay, excuse me. The first thing we're going to do, well, here, first we're going to convert the rep, well, and compound, that's like the treatment column, to a factor. So that's what this line does, mutate across rep, well, and compound. So apply the same function to all those columns, apply the factor function to those columns to turn them into factors. Control Enter. Here, head, we're going to just look at the first few rows and see what it looks like. Here we have, in the true experimental design, they looked at different concentrations of the compound and also different strains of the mold. I think, yes, for this example, we are just looking at one strain and one concentration for simplicity. So here are the reps, the wells all have numbers, and compound is the treatment we're looking at. It's either A, B, or A plus B. And here are also for simplicity, we're using 5-hour intervals, although the real data was measured at a much finer interval. And our response variable, OD, optical density, it's a positive continuous variable. Let's take a look at the data and make a plot to see what we're dealing with. The X variable is called hour in all caps. That's the name of the column in the data, hour. What we want on the y-axis is OD, our proxy of biomass. And we're going to make draw a time series, a line for each well, and we're going to color that by which compound it is, and group interaction compound rep and well. So each well within each replicate plate within each treatment is going to get its own line. So we're going to plot basically all the raw data on a plot. And I've also used this stat summary line to show the median for each treatment, each of the 3 treatments, as well as the individual wells. Control Enter. So here you can see there's not a huge difference in the medians between the 3 treatments. They mainly have this lag phase here, right, where they didn't grow much for the first while, and then they start to increase. Also, you can see that the optical density is never quite 0, right? Because the medium that the cells are suspended in is not completely transparent. It blocks some of the light itself. So whenever you're converting from optical density to biomass, you have to kind of subtract off that minimum value because because the medium itself is actually blocking some of the light, and then that allows you to get a closer correlation of the biomass, if that makes sense. And we are going to log transform the optical density as well. Oh, by the way, this plot has a log scale. I put a log scale on the y-axis because we want to look at relative changes in biomass over time. So that's why we're using a log transformation. But again, we're going to adjust that by subtracting the minimum off of it. So we'll take the log parentheses of OD minus the minimum of log parentheses OD. And we will calculate that minimum separately for each well, just in case it's slightly different minimum from well to well. And that's going to be the response variable that we are going to use in our model. So we actually are pre-transforming the data before the model, but which I usually say not to do, but for many cases where the process is really occurring on a log scale, it is acceptable to do that. So I'm going to Control Enter to do this adjustment. Now let's just plot that to see what the adjustment did. So we're going to use adjusted underscore log underscore OD as the y-axis. Other than that, it's the same plot code as last time. We're not putting a log y-axis because the variable is already log transformed. Here you see that the minimum value is 0. So Gompert's equation has to have a positive value. So this is good. This is what we want. We don't want to have negative values in our response data here. So far, so good. So now here, what I'm going to do is, before I actually fit the Bayesian model, I did want to get some information about what I would use as plausible mean values on the prior distributions of the nonlinear parameters. Now, I kind of cheated a little bit to do this because I am not a microbiologist. I have no clue what our plausible range of values for the A, B, and C parameter of a Gompertz equation. If I knew that, I would probably go, I'm lazy. I could have gone into the literature and found those numbers. But I didn't do that. I just quickly fit a frequentist version of the nonlinear model to the data and got the parameter estimates there and used those to determine the priors. Now, you could argue that's a little bit like double dipping. You're not supposed to use the data to determine the priors for the model. Yes, it's true, but I kind of set very wide standard deviations on those priors to kind of mitigate the double dipping aspect. That's not perfect, and really a best way to do this would be to get those prior distributions from the literature. But if you have strong enough signal in the data, the exact details of the prior won't matter for your final answer anyway. It will mainly be reflected in the efficiency of the model and how fast it gets you to the answer. So yeah, that's kind of like what I tell myself why it's okay to proceed this way. So yeah, the non-Bayesian way to fit a nonlinear least squares regression line is the NLS function in R. And the response variable is adjusted underscore log underscore OD. And here is the Gompertz equation, right? And the data is bioscreen is the name of the data set. And you have to put in start values for this algorithm to work, which I kind of figured out by trial and error. I'll save you that pain. Hit Control Enter to run this. And here we get basically the A parameter is around 3-ish, the B parameter is about 20, and the C parameter is some low number, 0.06. So we're going to use those values to figure out what are appropriate priors to put on the nonlinear parameters. Okay, so now, but I also, this isn't really the full model anyway. It doesn't account for the effect of treatment on those parameters. It's just looking at overall what generally we would expect these parameters to be. So again, I didn't like fit the whole thing to get priors, just a very simplified version of it. Okay, so hopefully that whole thought process makes sense. Now we're actually going to go ahead and fit the nonlinear mixed model. Now remember, whenever you have a model that has more parameters than just intercept and slope, or if you have like, we did this for when we wanted to fit different fixed and random effects on the different parameters of gamma and beta distributions, we had to use the BF function to create a multi-line formula. And you have to do this as well for when you do a nonlinear equation. The first line of the formula is the nonlinear equation, and then you have to have a separate line showing the fixed and random effects on each parameter of that nonlinear equation. So let's build that up piece by piece. First of all, our response variable is this adjusted log OD. That's our proxy for biomass again. And we're going to put together the Gompertz equation, which is A times exponential of negative B times the exponential of negative C times time. That's the Gompertz equation. We've just written it out explicitly here. Now, then we're going to say, okay, I think that this pyrosidine compound is going to affect the ultimate asymptote, the total amount of biomass that's going to grow for each of these, in each of the three treatments. So we're going to say we have to make a separate line because A is the parameter that's the asymptote. So we're going to say, okay, the asymptote can vary as a function of treatment, which is the compound, is the name of the column for treatment. And we're going to put a random intercept for each well within each replicate plate. So 1 by well colon rep. So we put a fixed effect of treatment and a random effect of well, basically for the different individual wells, since we have multiple time points within each well, on our asymptote. But I've said B tilde 1 and C tilde 1. Just for now, we're going to say our lag phase length and our growth rate is not allowed to vary by treatment, because for some reason we are more concerned if the final amount, the total amount of growth is more or less. That's often what microbiologists are interested in. They might not care quite as much about how fast it grows. It's more about ultimately how much mold is going to grow under these different treatments. And you have to put NL equals true to tell BRMS this is a nonlinear formula. Okay? And we're going to put priors on a bunch of these things. What priors are we going to put? Well, first of all, right, because remember that the asymptote parameter is around 3, which actually we wouldn't even need to fit the model to see that. You can see on this plot here that the asymptote is about 3, right? The maximum value is 3-ish. So I'm going to put a prior for a mean of 0 and standard deviation of 1 for class equals B, meaning fixed effects, and NL par equals A. So for fixed effects on the asymptote, I'm going to say the treatments have the prior on the treatment effect has a mean of 0 and standard deviation of 1, because it seems like a standard deviation of 1, a difference of 1 in the asymptote is a big difference. Thanks for being part of this, Elisa. See you soon, hopefully. So right, because the asymptote is in the neighborhood of 3, a mean of 0 and standard deviation of 1 is a reasonable prior. And I'm going to put a prior on the intercept for that asymptote as well, with a mean of 3 and a standard deviation of 1. Because again, I used my a quick look at the data to see that 3 is kind of the neighborhood where this asymptote is going to end up. And because the B parameter was about 20, I've put a prior with a mean of 20 and a standard deviation of 5 on that parameter. Class equals B for fixed effect, nonlinear parameter B, and coefficient equals intercept. Because it's going to be somewhere around 20. I put a standard deviation of 5 because that's pretty big with relative to 20. And for C, because it was a very small number, I've put a mean of 0, standard deviation of 1, which seemed conservative enough. And I've added some extra iterations. I've added 3,500 iterations with the first 1,000 being warm up. So that's 2,500 times 4. That's going to be 10,000 posterior samples, because this is kind of a more complex model with a lot of parameters. We may need more samples to fully characterize the distribution. So that's it. I've put together the syntax for the nonlinear model now. Does anyone have any questions on that? Can you give me a thumbs up if you're able to follow what we're doing here? Very cool. Even if you're not, we can hopefully clarify once you see the output of this thing what is going on. So now I've got my cursor somewhere in this huge monstrosity. I'm going to hit Control Enter to run the lines of code. And again, it didn't detect any change in the code. It loaded the prefit model object. The actual thing would take quite a long time to run, so do not try to run this right now. Let's look at the posterior predictive check plot to see if the distribution of OD values predicted is similar to what we observed, to make sure the model is giving us reasonable, just a quick sanity check to see if the model is reasonable. It's decent-ish. You see that the data have a bimodal distribution, which corresponds to that S curve, right? There's a lot of values near 0, not a lot in the middle, and a lot near 3, where it levels off. So our data, the black line, have this 2 humps. Our predictions also have 2 humps, although we seem to be under-predicting a little bit of the zeros and over-predicting this middle part. Maybe you could, so it's like, well, if you're theoretically tied to using the Gompertz equation, you might be like, well, that's not perfect. Maybe you could try other functional forms, but this is a very highly constrained equation, right? It imposes a very strict pattern on how we think things are going to go. So this might be close enough for you, and it might not. If you really wanted to, you could try other S-curve-like equations instead of Gompertz equation. And I think I did do that in the homework exercises, gave you another one to try out. Before we go to make predictions, I mentioned to you that for simplicity, we said, well, we were most interested in the effects of the treatments on the ultimate amount of biomass that can grow, so the asymptote. But we could also ask, well, does the lag phase time and growth rate also vary by treatment? We don't have to put, up here we put 1, that means just an intercept is fit for the B and C parameters. We could put fixed effects and random effects on them too. I'm going to go ahead and put a fixed effect of treatment on the B and C parameters as well, the lag phase, length, and growth rate parameters. Compound, B tilde compound, and C tilde compound. Other than that, the code is the same, and I've called this bioscreen Gompertz fit more pars because it's got more parameters. I'm not going to throw random effects on that B and C as well. That would be like putting a random slope model. I think, if you look at our data, we see there's not that much variation between the wells. So just having a random intercept is going to be plenty. Otherwise, you're starting to get into overfitting territory. And I've got some extra parameters now, or extra priors, because not only do I have to put a prior on the fixed effects of the asymptote, I'm also going to put a prior on the fixed effects of the lag phase and growth rate parameters. And I've put a pretty tight prior, a mean of 0, standard deviation of 1, because if you go back, remember, these curves look fairly similar. It doesn't seem like there's huge differences between the treatments. So we're going to just put mean of 0, standard deviation of 1 parameters on the B and C parameters as well. I'm sorry, priors on those parameters. So I've just given you that code there. So now each of the 3 parameters has a prior on the intercept of it and a prior on the treatment effects. We've got priors on all of them. I'm going to hit Control Enter to run this line of code. So the only thing I've changed is I've added those extra priors and I've added fixed effects on the other aspects of the curve. Okay, that runs, and I can do a posterior predictive check plot on that one as well, which I will do on line 202 here. Again, it looks about the same as last time, so it seems like whatever discrepancy was not resolved by adding those extra fixed effects. So it could just be a shortcoming of trying to impose this very strict theoretical Gompertz equation on something that doesn't necessarily follow it perfectly. Because yeah, if you go back and look at the data, there's not really this smooth S-curve. It's like very flat and then a very sharp turn. So it seems like the Gompertz is having trouble with this kind of kink in the curve, which you might need to make a more complex nonlinear model to truly fit that, if that was something you cared about. But since we really only care about this final asymptote anyway, it may not matter too much for us. Now, as we have done already several times in this workshop, when we have different models that we fit to the same data set that have different complexity, we're going to use leave one out cross-validation to generate an information criterion to say, was it worth it to add those extra parameters? Does it improve the model fit enough to justify the additional complexity? We've done this already, I think, pretty much every week. We're going to do it again. Add underscore criterion is the function we use to compute the information criterion based on the leave one out cross-validation for the model. And LOO or LU is the name of the criterion. And we're going to run that. I'll highlight this and run that for both the models. Again, it was pre-computed by me previously, so if you were computing this from scratch, it would take at least a few seconds to calculate that. Then we'll use the LOO underscore compare function, leave one out compare, to look at the difference between the models. And we see quite a big difference. The model that's more complex with more parameters is a much better fit. The difference in the log posterior density is about 80 units with a standard error of only 18. Oh yeah, and I think it was Brad who asked last time about a rule of thumb about what constitutes a big difference. I was looking that up, and there is a publication in the literature by the people who developed this metric that says a difference of 4 is a decent cutoff for, are the models similar or different? And we have 80, so it's like, well, they're very different. And the way that you calculate a p-value for this is by basically dividing the difference by its standard error. That gives you a z-score for the difference, and then you can calculate a p-value off of that z-score. And if you know z-scores, you know that a z-score of 2 plus or minus 2 is kind of equivalent to a p-value of 0.05. So like this z-score is about 80 divided by 20. It's about 4. So it's going to be significant if you go off of p.05 as a significance cutoff. That's what you would use to convert this thing into a p-value if you wanted to do that. So yeah, it fits the data better to add these extra parameters. that allow the growth rate, the slope of the curve to be different between the treatments. Even though to me they look pretty close, apparently it's still different enough that that there's some variation between the treatments that's being picked up. So now the remainder of the code here is to, now that we've decided our model with extra parameters is better, we're going to use it to make predictions, make plots of the posteriors, compare the parameters between the treatments, and test hypotheses about whether the treatments are different or not. How are we going to do that? Well, again, EM means isn't really apt for this kind of data. So we're going to have to create a prediction grid again of all the treatment and time combinations we want to plot, generate posterior predictions for each of those combinations, and plot them. That's what we're going to do. So we're going to use the expand underscore grid. And first hour 0:120. This is basically just for plotting purposes. I'm going to predict, make a model prediction for each hour from one hour apart from 0 to 120. It doesn't really matter how many you do. I just want to make a smooth line of fitted model prediction. So you could do it by tens if you wanted to like save computation time, but I'm just doing it by ones. And then each combination of time and treatment, which is the unique values of compound, that's my prediction grid. And then I'm going to use the function add underscore epred underscore draws, which means generate fitted values from the model for each of these treatment combinations from our model, setting random effects to 0 because we want to make average predictions, averaging over the random effects. And I'll do n draws equals 1,000. Remember, we had 10,000 posterior draws, but just to make a plot, that would be way too many rows in our data frame. It would take a long time to compute. So I've just cut it down to 1,000, which is still plenty for plotting purposes. Control Enter to run that. Yeah, so you can see it already has 300,000 rows, right? We have 120-ish time points times 3 treatments times 1,000 draws. So, it would be 10 times that if we did the 10,000, which you can still do. And I would do that if I was publishing it in a manuscript. I would just use all the draws, but just for now, we're just doing 1,000. And I've made 2 versions of the plot. One is just the fitted model trends, and one I've also included the raw data on there as well, just to compare and contrast. So the first plot, the x-axis is the hour, which was in capitals in the original data, so it has to still be in capitals. And .epred is the name of the column that is by default generated when you're drawing expected values from the posterior. And we're going to make a separate line plus a credible region for each treatment with a colorblind friendly color scheme. I'll run that line by hitting Control Enter. So here's the fitted. So yeah, these are the predictions for the average well that is in the B compound treatment. It looks like on average you end up with a little bit higher biomass under B than you do with A or A and B combined. And it seems like the A and B is right in between. So there's not a lot of interaction between the compounds. They kind of have an additive effect, it looks like, at least in this example. And you have to squint to see it, but I've made different levels of shading for the 50%, 80%, and 95%, which are the default credible regions that are drawn by this function. You can change the defaults if you want to. That looks very clean and nice, but sometimes I like to show the data as well. If you're putting this in a paper, the model, again, is just an abstraction. The data are the real observations. We should plot them as well. just to A, to show the variation that exists, and B, to help reassure people that our model is a decent fit to the data, that it corresponds to reality. So that this next line of code, which I provided you, I'm going to run that, and it'll make the same plot with the raw data on it too. So yeah, not quite as clean, but we do see a little bit of the variability that exists. While we still see these overall average differences, we see that there's actually quite a bit of variation, around it. And there's some overlap, that you don't see if you just plot those perfectly clean model predictions. Your mileage may vary on what you prefer there. Last couple of things. Okay, there's our plot. What if you wanted to make formal statistical inference about the difference, the effect that treatments have on the different parameters? Well, you kind of have to do that separately for each parameter. Like, does treatment affect the A parameter, the B parameter, or the C parameter? Remember, A is the height of the S-curve, the asymptote. B is like how much lag there is, and C is how steep is this slope here. So we're going to do EM means, but luckily EM means actually supports nonlinear models in VRMS and will give you means for each of those parameters for each treatment, which is kind of neat. And my EM means formula is pairwise by compound. That means I'm going to generate the means for each compound as well as the difference between each pair of compounds. And if you supply the NL par argument, it will say which parameter do I want the means of. So I'm doing it for A, B, and C. Every time it's pairwise by compound, NL par equals, and each time I've done a different one, A, B, and C. I'm going to run this code here to do that. And then this next bunch of code is to get the credible intervals on our estimates for each parameter and the credible intervals and Bayesian p-values for our estimate of the difference between each pair of treatments for the parameters. We've done this every time so far. It's just that we have to do it not just for means, we're doing it for 3 different parameters. So that's why there's 3 lines of describe posterior for each. 1 each for the means of the 3 different parameters, and 1 each for the pairwise treatment differences for the 3 different parameters. Describe underscore posterior every time. And yeah, the code is the same for each of the lines. It's just that I'm using EMMA, B, and C. EM means is the name of the object within that has the means. And contrast is the name of the object that has the contrasts. And our CI underscore method, we're going to use an equal-tailed quantile credible interval, which I think is fine for most uses. CI, I'm going to use the same widths that we had in our in this figure just for consistency, 50, 80, and 95%. I'm not going to do p-values on the means themselves because we're not testing whether those are different from 0. We're testing whether the difference between the treatments is different from 0, right? And so we will put contrasts here to get the posterior distribution for the contrasts. Test, we're going to do the 2 different styles of Bayesian p-value that we've been looking at so far throughout all of the examples. We're going to do them. So I'll hit Control Enter to run this entire block once I've done those 6 lines. Okay. Now we can look at the values by just printing the things. And remember, like these A ones should be around 3 for each treatment. Maybe the B is a little bit higher than AB, which is a little bit higher than A. I'll hit Control Enter line by line. You can see that. So yeah, here's the value for B is about 3.2 versus 3.06 for A. credible intervals accordingly are pretty narrow. 95% CI between 3.1 and 3.28 for B, for example. It's kind of confusing because there's like A, B, and B are the names of the treatments, and A, B, and C are the names of the parameters, but hopefully it's not too confusing. So this is the asymptote. Now for the lag phase one, it's quite similar for each of the three. There's about 22 to 23 lag phase, which you can see on the plot. The thing is flat till around a little past 20, so that makes sense. And I've had to add this extra argument, print EMMC post digits equals 4, because by default, it rounds things to 2 or 3 digits when you print these objects, but the C parameter is so small, it was like 0.06, that you can't see the differences between the treatments unless you go out to 4 decimals. So I just had to add that argument so you can see that. And here they are. So here's the growth rate, the slope of the curve. It's around 0.06 for all the treatments, but maybe at the third significant figure, there's a bit of a difference. And now we can also look at the pairwise treatment effects too. Again, we're looking at 3 different things. So like, you wouldn't necessarily have to present all this in a manuscript. It depends what biological hypothesis is most interesting to you. Are you caring about the effect on the growth rate or the effect on the total amount that grows, carrying capacity, and so forth? But I'm showing them all here in case you're interested. So we'll look at the contrasts for the asymptote between the treatments. They're relatively small differences, where the mean is 3, the differences are about 0.1. The credible intervals are close to 0, but we do have modest evidence of differences between the asymptotes. For A and B, for example, the difference here between like this green line and orange line where they flatten out, at least the probability of direction is 98%. So that means there's like we're 98% sure that difference is bigger than 0, basically, or smaller than 0 in this case. And so, there's a little bit of evidence for a different effect of these different compounds. For the lag phase, we see pretty small differences where the uncertainty is big, so we don't have much evidence for a difference there. For the growth rate parameter, we have actually, despite the fact that those absolute differences are pretty small, we have pretty good evidence of a difference in the slopes, basically of the slopes of the middle parts of these lines between the different compounds. That could be biologically interesting. I don't know. Like I said, I'm only a statistician, not a microbiologist, so I don't know if those numbers are meaningful, but maybe some microbiologists here can shed light on that. Anyhow, that is basically it. I've included, as always, in the slides and in the text version of the lesson, I've included text that you would include in a manuscript to write this up. And here I'll just show you a figure that I made that I thought was like a pretty decent figure that could go in a manuscript. It's basically what we already just saw with nicer labels and everything, and the legends moved around so they're in a nicer spot. That's it's not too different than what we already plotted, but I think that would be a good way to, again, I've shown the raw data and kind of grayed out so that it's not too distracting, but you can see the variability, and then here's the model predictions as well. Because the model is only useful if it is a good fit to the data, which this one is more or less. Yes, well, here, I'll say a few more wrap-up things, but does anyone have any questions about this final example? I'm impressed that you are all still, or that at least a lot of you are still here. So let me just go back to the slides and see if I have any wrap up stuff here. Where are we? Yeah, I wanted to do this every week, but we always ran out of time. I think it's nice to kind of like just verbally say what we've learned each time, because it helps you remember what you have learned and learn it better. And it's kind of nice it makes you feel good to know what you've learned. So yeah, what did we learn just today? Well, we learned about what to do if you have ordered categorical response data, what kind of model to fit, and how to use that model to make predictions and compare treatments in different ways. And we learned about nonlinear models that you could fit if you, for example, have microbial growth data that you want to test different theories about how they grow and how different environmental conditions or treatments or whatever affect that, and in a Bayesian framework. So that's pretty cool. I congratulate you all on having made it this far. And that's basically it. I just want to mention, in all the lessons at the bottom, not only do I have little homework problems if you want to do them, I've also provided a few links to books or tutorials online and so forth, for further reading on these topics, since we really have only a lot of times scratched the surface. So yeah, you should definitely take a look at that. And again, I'll put it in the chat one more time. Please do fill out the feedback form, questionnaire thingy, so that I can get feedback from you. Yeah. So I'll just stop screen sharing. I did want to, yes, and I see Grant has come on the screen. Let's all thank him again for all the help he's provided. And yeah, I really appreciate it, Grant. So thank you. Um, I just want to end by asking you all a couple questions. There's no obligation to answer any of them, but I am curious, just for my own knowledge, how many of you would consider using any Bayesian techniques that you've learned in the last few weeks in your own work? I'm just curious if anybody has a specific data set or project that they're thinking about would be good to use Bayesian techniques for. If anyone has one, I would love to hear about it.
So I'll jump in, Quinn, and you know some of my data before, but
Yeah.
for the rest of the group, I'm a biocontrol worker who host range tests insects as for safety to use in environmental out in the environment to fight invasive weeds. So the setup of our experimentation is that we hope and expect to get tons of zeros where the insects do not
Yep.
feed or oviposit or survive on non-host plants, but they do really well on host plants, and occasionally will do minimally well on some very close relative. So
Yep.
primarily a binomial distribution. And so the work that we were doing, particularly back with the binomial, is very interesting to me. You and I had talked about this in prior times. So when I saw the course offered, I thought, well, this is a good chance for me to make some sense of what you were trying to tell me. So for me, I think it was very useful. I'll probably still have to beg for help, but at least now I understand what I'm asking for
Yes,
when I
and
get
yeah,
there.
and a well-informed request for help is the best kind, right? So yeah, and I think for your stuff, it's really going to be good to use Bayesian techniques, because if you do have like the non-host, if you observe in all zeros for that particular plant, like for that treatment, the frequentist, like maximum likelihood approach it fails to calculate kind of you can't get an odds ratio of the difference between all zeros and something else, right? Because there's no
Right.
variance in that one that's all zeros. The only way you can get one at all is by putting in some priors to constrain things a bit. So yeah,
Right.
for those type
Well,
of
and my
things, it's very nice that, but the numbers you get are kind of sensitive to your prior. So
sure.
whenever you do this stuff, definitely try out different priors and see how sensitive your results are to them, and maybe even in a publication show the variation across a few different choice of priors, because that's real important.
Yeah. Yeah. And when I did it earlier, when I was doing a much smaller data set with parasitoids, I actually came up, and you and I've had this discussion, but I came up with a transformation that allowed me to still use a
Yep.
frequentist approach,
Yep.
but that was because my data set was exceedingly small.
Yep.
In the data set that I'll be working on now, I have 88 plant species, something like 3,000 iterations,
Okay.
and and 5 variables. So it's just massive, and I would never be able to do the transformations even if I wanted to stay frequentist. So this is
Yep.
again
Yep.
really appropriate for helping me out
And like we
in
talked
moving forward.
about, your transformation that you were doing was kind of like a prior. It was kind of like a
Yep,
crude prior. So
yep,
if you're going to do that anyway, you're already putting in prior assumptions. So why not do it in the more mathematically rigorous way of a Bayesian model?
sure,
At least that's my thinking. If you're going to do that anyway, you may as well
yeah.
in for a penny and like
I,
go the full
I,
Bayesian route, you know? Yeah.
and I don't disagree with you. My only defense for myself is back when I was doing that first paper, I had never heard of Bayesian
Yep.
statistics,
Yep.
and I didn't even know there were models out there to help me do
Yes.
it. So I wrangled my own way through. Like you said, I had to learn a lot about Bayesian statistics in order to make it work, because I had to have priors and posteriors and the whole thing. So it worked out well in preparing me for this. But um, so baby steps.
Yes, and I don't want to sound like dogmatic. I mean, different tools can give you the right answer in different ways. So it's not like Bayesian is always the best or anything like that. It's just for your particular case, it seems like it lends
Yeah,
itself to it. So yeah, well, thank you. Yeah, we'll definitely have to revisit that in the future.
appreciate it. Thanks much.
Yeah, anytime. Anybody else? It's okay if not, but I was just curious.
Yeah, Quinn, Eric here.
Hey, what's up, Eric?
Hey, so yeah, I know you know this, but I'm obviously going to be trying to do this in SAS with that count data that a lot of my studies revolve around. We'll see how that goes, but if not, I'm going to have to learn this in R, probably using the code from this workshop as a skeleton for how to do that. So,
Yes, and I mean, yes, we've only done, R, we're really using Stan, but R is just kind of like the interface we're using to work with Stan. But yeah, I'm kind of embarrassed at how bad my SAS coding is. But there is like ways to do pretty much everything we did in SAS, although to be honest with you, it is quite cumbersome to do some of the stuff we did in SAS. Like yeah, but it can be done, and yeah, at least certainly any of the models we fit can be fit in SAS using proc MC. But like a lot of the like post processing with like quick and easy code to compare treatment means and make plots of it and stuff. Yeah, that's going to be very tough to
yeah.
do in SAS, or maybe somebody wrote macros to do it, but I don't know about them because I'm not like a real SAS head. But yeah, certainly the models, obviously there's no reason why you couldn't fit them in SAS. Yeah.
The models are really all I
Yep.
care about, just so
Yep.
I can see what the final
Yep.
results actually are. Because
Because yeah.
as you've shown me, the frequentist view is overestimating the mean effects of my treatments quite heavily, and
Sometimes it can. Yeah.
it needs to be correct if I'm going to report those results. We'll see what happens when I get another year
For
of
sure.
data. But anyway, thanks for the workshop.
Yeah. Yeah. Anytime. I actually took a Bayesian class in SAS last year that with some of the training credits that the old era statistician had. It's pretty good, and they do a good job. It's kind of like what we just did, except in SAS. I would recommend it, but it's like, you'll have to shell out, 1,500
Yeah.
bucks or so
Yeah.
to do it. But it was actually pretty cool. Yeah. Great. Does anyone else have any other questions, comments, anything like that? Well, great. We may end early for once here. Well, yeah, just I won't keep you too much longer, but I'll just say that I appreciate you all being willing to learn, and it's been fun, and hopefully we'll do another similar thing on a new topic soon. So if you do have any other topics that you want me to make curriculum for, or if you have an existing tutorial or something that you want me to like teach off of, definitely email me about it or throw that into the feedback form and we'll take a look. Yes, and again, I'm gonna post all the. Oh wait, before we do go, I'm sorry, I just want to say something logistically because Adam asked about this last time, but I just want to repeat it one more time. You cannot, after this class is over, I am going to probably within the next week or two take your usernames off of the SEA stats training account on Scinet. All that means is that you cannot run jobs under that account anymore. But if you use Scinet at all, you probably have your own project account. You can run jobs under that. If you don't have one, you just have to go to a form on the website and apply for one, and you should get it, pretty much as a matter of course. You just all it means is they have to allocate like some space on the server for your stuff. And that's one. Number two, all the data that the example data that you copied into your own space was copied into a space called 90-day data. As the name implies, it automatically deletes anything that was not used in the past 90 days. So if you do want, if you like saved any of the worksheets with like your notes or anything like that, you'll want to copy those to your home directory on Scinet or export them or anything. Does anybody care enough? Like I can walk you through that process now if you like. Would anybody like me to do that? How to copy or move the files to a different spot?
Just a clarification question on that.
Go for it.
With the files that we were using that are like the filled in ones that you had, is
Yep.
the website for this training or this workshop is still going to be up
I'm going
indefinitely,
to keep those up there.
correct?
Yes.
Okay,
all of that will still be there. So like you don't need to get those off of there. Yeah. So here, let me just
that's why I was asking
share
that question.
screen again one more time and just show real quick because somebody has said they are interested. Yeah. So basically on this workshop page, that's always going to be there. The worksheets are there. So you don't need to get them off of Scinet. And same with the slides and the text version. All of that's there. But let's say you did have a file on Scinet, a file or two that you wrote notes in and saved on the server. You would want to go, like, let's say I saved this worksheet with notes that I put. I would just go to here are the worksheets, right? So let's say I wanted to export all of them. You would just select them all, and then you could export them by going to this gear icon and clicking on the export button. And then it will just download to your downloads folder a zipped file of all those. So that's one way to do it. Or you could, I mean, that's probably the way you'd want to do it. You could also move them to another location on Scinet that's not inside of 90-day data. But like you can use your home directory, for example. I could move them to my home directory on Scinet in that way. So if I went to the tilde is the home directory, I could move or copy them to the home directory. And then they would not be deleted after 90 days. So that would be how you do it. But you can go back into the Scinet server and run RStudio server jobs and still work with the example data that's in 90-day data as long as you want to. It's just that you won't be able to use the SEA stats training account on it, but it doesn't really matter. You can just you can run it off your own project. Scinet wants to keep bookkeeping on what Chris projects and that kind of thing are using processor time. So yeah, that's that. So that hopefully that clarifies things for you there. Yeah, so I will stop the recording now, but again, thank you all so much for being part of this, and please fill out the feedback and shoot me a email or call if you want to do Bayesian or any other kind of analysis on your own data. Thanks.